Okay, our final talk is going to be by John Barford from Hong Kong University of Science and Technology, and he's going to give a talk on a novel metabolic pathway optimization method.
Okay, if I could start by just thanking the organising committee for allowing us to present the work at the conference. The title of the talk is A Novel Metabolic Pathway Optimization Method, and it basically was part of a graduate work by Kelvin Wong, who's sitting over here. I'm rather pleased he's sitting over here in case I get some difficult questions from the audience, because then I'll be able to ask him to help. So as part of his work, it's also part of developing a metabolic engineering course here at the university. And when I started that process, I realized how little I knew about that area. I'm not so sure I know very much more about it now, but in the process, we got to look at the basis of metabolic engineering, the mathematics of it, and actually think about whether it was a lot of the things that were predicted mathematically could actually be implemented in a practical sense. So the talk really is about what sort of analysis is done, and then whether the outcomes are sensible and able to be practically implemented. Okay, so as you know then, if we want to try to produce things by microorganisms and then subsequently try to either improve the performance or optimizing it, we generally do that in one or two ways, either manipulating the reactors and the operating conditions that they have, or the second one is to modify the cellular metabolism by employing some techniques, and obviously metabolic engineering or genetic modification is one of those. And so basically, if you're trying to produce a desired product, you may want to do, there's a whole range of things you might want to do. You might want to increase the yield of this product. You might want to minimize something that you don't want. You might want to introduce new pathways, etc. You might want to extend the substrate range. And it's all that essentially will be carried out by modifying the enzyme activities or the quantities of them generally. Okay, so this is the sort of summary of metabolic engineering in the sense that obviously we need a series of information for things of the cell, details of the enzyme kinetics, for example, then go through a process of analyzing that under some experimental condition. You then design some strategy for improvement, and then you implement that and start again. It's a sort of cyclic process. And what we want to concentrate today is these two here, where we assume we're having some metabolic information available to us, and then we talk about how to actually analyze it, and then subsequently how to improve the process, and then question whether it's implementable or not. Okay, so the idea is generally to identify the enzyme and the pathway that should be modified, and often it's more than one. And the general outcome is an attempt to increase the flux. So for a practical solution then to a strategy, you want to be able to do two things. You want to firstly identify which enzymes you want to modify, but then secondly you want to estimate the magnitudes by which these enzymes need to be modified. And the classic metabolic engineering approaches do the first one reasonably well insofar as they can show you which enzymes are causing problems in terms of holding up the flux. But they don't do the second one all that well in terms of showing you what strategy can then be actually developed and then possibly implemented. So the idea of this algorithm then is to really confirm the first one, which is done by most processes, but then to really concentrate on trying to get a practical outcome that can be actually implemented. Okay, so there's a number of different optimization processes available. The classic one in metabolic engineering is the metabolic control analysis theory. So that's well established, and will certainly identify, if you have enough information available, will certainly identify which enzymes are causing problems. The second one is just a simple flux theory where if you increase all of the enzymes in a particular pathway by the same amount, then you can generally get the flux increased by that method. The third one is a whole sort of mathematical series of programming which can be used. So basically they involve some programming and some defined objective functions. And this is part of the sort of strategy we're going to talk about today. But the first one is also included in the analysis. Okay, so there's 2 or 3 groups here then which have attempted to optimize pathways. And I don't want to go through the details of what they've done, but simply just give you some idea of their approach. Okay, so this approach here is where they increase the flux by having all of the enzymes in the pathway, which is part of that flux, to be increased. The second group here is basically using various mathematical techniques to do the same thing. But what it does, it's just in the original MCA analysis, what happens? We can in all of these methods we can identify which enzymes are contributing to the problem, but we don't get any. We don't say, it doesn't give us which one should be modified and to what extent. So there'll always be one enzyme that has the highest flux control coefficient, for example, but the other enzymes can also be having an effect on the flux, and so you don't get any information about which ones are changing by how much. This is another approach here, which is a similar one, basically on linear programming. And what we'll do is then take these 3 established optimization techniques and then compare them to one which we did, which I'll describe in detail in a moment. Okay, so the basis then of metabolic flux analysis is trying to understand how sensitive the flux variation is to changes in various enzyme levels. It's carried out generally at steady state, and there has to be some experimental information. So we have to know something about the kinetics of the enzyme, and once we do that, we normally incorporate that into some mathematical methods, generally some metabolic modelling. And often that modelling is then used to extend the application of the theory to a larger extent. So when we measure a whole series of experimental measurements, we don't want to have to do this over and over again. So we normally have one sort of starting point where a massive amount of information is gathered experimentally. Then we tend to use mathematical methods to help us sort of develop the optimization further. Because if each time we have to make these measurements, it's quite a considerable amount of information to get. Okay, so this is the basis of the theory, and again, I'm not going to go through this. This is quite well established. It's just talking about, for example, flux control coefficient here. The second one is just saying how much the flux changes with a change in enzyme concentration. So it's a simple thing, and this is the term which eventually we want to get for all of the enzymes in the cell to identify which ones are causing the major problem. So there's a whole range of these other things defined here, some of which can be measured experimentally, and some of which can't be easily measured experimentally. At the end of the day, what happens is that basically the information that is known is put into one matrix, and the information that is not known is put into another, and the relationship is such that we can solve the matrix to find the information that we want, which is generally the flux control coefficients. So the object of the metabolic control analysis is then basically to find out which enzyme has the largest control of the flux and possibly presents a bottleneck in the process. Okay, and that's generally identified by finding the one with the largest flux coefficient, and that's a relatively straightforward thing as long as we know the kinetics of each of the enzymes and have some measurements at one steady state of all the intermediate concentrations, etc. So the initial identification of where the problem might be is relatively easy, but there's 2 problems that come from that. The first one is that all of the enzymes can affect the flux. So the one you identify as being with the highest coefficient is not necessarily the only one that has a considerable effect on the flux. And then secondly, if you then decide to change the enzyme concentration to overcome this problem, the question is, well, how much do you need to change it by? And whether there's any limits to the process. So if we keep increasing the concentration of the enzyme which is limiting here, we eventually hit the wall and we don't get a further increase in flux. So just increasing things, if you like, in a random fashion and hoping that the flux increase is large is not necessarily going to be successful. And this shows you the concept I just said. Basically what this is showing you here is that there's an expected increase in flux with an increase in enzyme concentration, and then there's an actually observed one, and those are quite different once we get out of a very small operating region at the bottom here. So if we change an enzyme by a relatively small amount, then what happens in actual case and what happens in your predicted or expected case are similar. But once we start going outside that range, we don't get the large increase in flux that we might expect with a large increase in the concentration of an enzyme that you've chosen as being the problem. Because basically what happens then is that the analysis that you carry out, the metabolic control analysis you carry out under one condition, if you change to another one, is not necessarily strictly applicable. And what happens generally is that the bottleneck shifts from one point to another, and you end up chasing yourself around pathways, moving from one enzyme to another. And if you were to try to implement that practically, you'd start by increasing one enzyme in the cell. Your second round, you'd increase the second one, and you'd just keep going around when you virtually hit a limit, because there would come a point where you can't simply produce any more enzymes inside the cell practically, because there's an upper limit to how many you can produce. So what you want to do is try to develop a method where you edge towards a optimal flux by allowing the change of any of the enzymes, but only do this in a very small step over and over again, which keeps you relatively in the region where you can expect the enzyme increase to have a relatively high effect on the flux. And this is done by having some iterative method, which we basically re-estimate the control flux after making a very small change in one enzyme, and then do the process again. It may indicate the same enzyme as a problem, and you change that a small amount as well. If it changes to another enzyme, you then change that by a small amount, and you keep continuing this process until you have what you might accept as an optimum. And hopefully by that method what you end up with is not too many enzymes being changed, and not to a huge extent which causes other problems in the cell. So that's the basic sort of concept we're trying to get across. So what we're going to do is compare three or four different optimization methods, and you'll see that they have quite different outcomes, which is a bit of a problem in the first place, because generally you don't expect there to be one optimum. And then secondly, you'll see that some of them end up with fairly impractical outcomes in terms of changing large number of enzymes, and some of them to such a large extent that you would have to question whether they would be practically possible. So now, okay, now I'm not going to go through obviously the detail of the algorithm. Only this is the sort of flow chart on the right-hand side. Having a bit of a problem here, so I can't see it from this side, and my eyesight's not good enough to see it from this side, so it's rather difficult to explain. But in general, okay, what we're doing is the metabolic control analysis is a well-established technique. So as long as we have some experimental results of intermediate concentrations, etc., in enzyme activities, and we have some description of the kinetics of the enzyme, we can undertake that analysis. And as I said, generally then, the enzyme with the highest flux control coefficient is chosen and the concentration is increased. It's just a matter by how much you increase it, and then you have to determine what the outcome is, either experimentally, which is tedious, or mathematically from the information you have before. And the second one is the most common one, because of the large amount of experimental information that you need otherwise. And then some hill climbing optimization technique is used so that we basically iterate, go from one step to another step to another step until we get to a final outcome. So at each step, something is identified and then changed. The second diagram here just shows you the same thing. In this case, though, we don't just choose the one with the highest control coefficient. We basically try to look across the enzymes to see what is the actual best value of flux against some objective. So instead of simply relying on the control coefficients. So we want the largest new flux at the steady state. And what I mean by that is try to summarize in this diagram here. We're basically, depending on how much we change the enzyme concentration. So, okay, we've identified the enzyme we want to change. We then say, well, how much we're going to change it by. If we change it by a small amount, we get a result which may indicate that the same enzyme has to be then increased a little bit more. Or if we increase it by a large amount, it might indicate and switch to another enzyme which needs to be changed. So depending how you do this, you can have different results from the MCA analysis. So the MCA analysis would indicate one thing, and a large step in a direction would indicate a swap to another, and they don't actually necessarily correspond. So the algorithm then is a little more active, if you like, in trying to hunt down the global solution across the cell. And so the whole enzyme bodies, each of the enzymes are allowed to be increased a small amount. We see what effect that has, choose the one that has the most effect, go on to the next step, and keep going and keep going. Okay, so to do that, to do the comparison, we then just chose what here is basically a model pathway, and it's one which is being used, so it's a glycolytical pathway from glucose to ethanol, relatively well established, and some optimization of these sort of pathways has been done before. Okay, so we're taking this as if you like, a model system, and we're now going to apply 2 or 3 different optimization techniques, 2 of the ones which have been done before, and the one that we've mentioned doing ourselves, which is the hill climbing. And then this is the result, and I hope you can see it. Again, I'm not sure. So yeah, I hope you can see it. So what the outcome of this then is twofold, and the most important outcome, of course, is the one at the bottom here, where you see the 2 other techniques, the ICOL and the Caxo, those workers. The amount of enzyme change there is very, very significant compared to ours. Okay, ours has a very small change in enzyme, total enzyme change. It's very small compared to these. So these are having outcomes which involve massive enzyme changes. And the other thing is that those 2 methods, the 2 other methods, indicate that different enzymes having to be changed and more enzymes having to be changed. So the outcomes of these algorithms are quite different in terms of indicating which enzymes need to be changed and to what extent. And these 2 in particular generally is to very large quantities of enzymes having to be made across the board. So this is just the detail of that, just showing you that they are different, so that the end results of these will indicate that different enzymes need to be changed. And then secondly, I said the more importantly probably, is that the extent to which they need to be changed are very considerably different. One of those suggesting the enzyme at least 3 or 4 times the original concentrations. If you start doing that to a large number of enzymes in a cell, that you're likely to hit some sort of a limit to whether it's practical or not. Okay, so this is the sort of, if you like, questions that arise from this. If you have an optimization method which is based on the mathematical analysis and some experimental work. And the optimization that we developed would indicate that we can get the same flux increase, but by modifying much fewer enzymes and to a much lesser extent. Then this is the question that follows. I mean, in the other methods, they're often modifying all of the enzymes, which clearly is more difficult to do than modifying one or two. And secondly, in the other optimization ones, the extent to which the enzymes need to be changed are very high and considerably different again from ours. Okay, so the advantage then of the, well, what we think the advantage of the algorithm we've developed is simply that it's a relatively simple strategy because you're using relatively well-established techniques in the control analysis problem, and that you're just doing it in an algorithm does it in a way which is small increments across the whole of the enzyme spectra, and then what happens is the outcomes tend to be that fewer enzymes need to be modified, and very much less enzymes have to be made. So that's, we think, kind of a suitable advantage. The limitations are, can the cell make small incremental changes in enzymes, individual enzymes in isolation, or does it have to change lots of enzymes at the same time and by relatively large amounts? So that is the feedback, if you like, or the control within the cell sufficient to be able to achieve what we've done mathematically? So and that question, I think, is one that really holds to all mathematical approaches in this area. So it's not just limited to ours, but the question does need to be asked, is whether what you do mathematically practically possible within the cell. The second one is that, of course, the amount of information you really do need for all of the interaction from one enzyme to another is quite substantial. But that limitation is, as I said, is faced by all pathway optimization methods, not just the one I've shown you here. So some of the limitations are, if you like, general and across the board. And the only specific one that I raise is whether, the small incremental thing we've done mathematically is possible physically in a cell. So this just says what I've said a number of times already then, that this method allows you to identify which enzymes should be modified, tells you by how much they should be modified to achieve a certain outcome. And the outcome generally for this method is that the number of enzymes is lower and the extent to which they have to be modified is lower. So that's really where it ends.
So our speakers have done an excellent job of staying on time. So I will allow a couple of questions here if people have them.
Can you imagine a form of your algorithm that kept the total enzyme concentration static, and as you increased one that was the highest effect, you decreased the one that had the lowest effect? And would that give you? Do you think it would give you a different optimum? Or?
Yeah, it probably would. I think when we first did this, let me answer in a different way. We also allowed, initially our thing is only to increase enzymes, not to decrease them. But it's very easy to implement either decrease only or a mixture of them, yeah. And I think, the question you ask is really what we've found, is that there's no point just increasing these enzymes without any constraint. So many of these optimization techniques had limited constraints. They just push a button, off it goes, and some output comes, and they don't really discuss then whether this can be implemented. So in our one, we put an upper limit. We say maybe allow the enzymes to increase by 20% maximum. We won't allow it to go any further than that, so there's no point coming out with outcomes that will make the protein load in the cell so high as to become impracticable. Right. Yeah, so the next step is, I think, what you just said, is that looking at basically different constraints. and probably better trying to understand the constraints in the cell first and then developing them rather than us just dreaming up strategies or constraints that are not real.
Thank you.
Are there any other questions? Well, I just have a really brief kind of question, and the question is very kind of a computer science question. Just for the experiments you ran, how long does it take to run them in terms of the actual computer time to run the algorithm?
Oh, it's very fast. Yeah, it's not a computational problem. It's more a conceptual problem how you put the thing together. So it's not limited by that.
So just using typical standard linear programming techniques and running the algorithmic running time of those are comparable to any linear programming problem.
Yeah.
All right. Thank you very much and enjoy your lunch.